So let's see, what time is it? We still are a bit early, so I'll just make sure I don't start ahead of time.
So nice chicken.
Yeah, and so this is something that's interesting for me, where I work as a research center in grad studies. We really are a global health, if you will. So biosciences, where it's not, although the definition has changed a lot, and it used to be where if you worked in a medical faculty, you could not, work on veterinary pathogens and the inverse. Whereas at my center, there was no real, veterinary pathogen versus human pathogen. So in working with E. coli, I have that opportunity to look at both the human diseases and also some important animal health diseases, both caused by E. coli. So, and they often share similar virulence factors. So I'm going to pretty much talk about that on both sides of things that interest me as a bacteriologist and an infection and immunity oriented person.
We also have some participants interested in veterinary, so it will be very nice.
Great. So I'm mostly molecular genetics and basic bacterial pathogenesis is my background, but I'm hoping I'll have a story that will interest some of you and teach you some different things. Some things you'll already know if you're in microbiology, at least for the introduction, but I think it's always important to re-emphasize some aspects of things about how important horizontal gene transfer is. And we just had and we're continuing to have a pandemic with this COVID-19 epidemic, but there's an ongoing pandemic of antibiotic resistance that is not going to go away anytime soon. And so because of this, it's important to understand bacterial pathogenesis and come up with different ways in some cases to fight against some of these pathogens that are becoming more and more difficult to treat. So that's my angle here. I have a chat I see. I don't know if we can. Okay, my knowledge of the Ukrainian letter system is limited, so I hope everyone is well. And for me, it's 8 in the morning, so I'm just starting my day. You guys have had you're almost wrapping up your day. I guess it's 3 p.m. or 1500 hours there, right?
Very hot weather.
You're having hot weather?
Very, maybe 30 around more.
Okay, we did have that here about 3 days ago. It was ridiculous for us to have I guess it was 29 or 30, but now it's down to about 22. It's a lot nicer here. So that's great. Okay, so I'll get started. So it's a pleasure for me to give this seminar as part of the International Summer School in Odessa. I'm very pleased and proud to have been contacted to do this. It's very interesting. You have a great lineup of speakers, and I'm happy to be among them. And so I've been working on bacteria for over 20 some odd years now, particularly bacterial pathogenesis, with a lot of work in enterobacteria. So, and some cases as well, other bacteria associated with poultry infections, things like Campylobacter and Salmonella. And so my main interest has been E. coli causing extra-intestinal infections. And so my title, Pathogenic E. coli outside the gut. So it's adapted to survive and thrive away from home. So from that idea, we're used to E. coli considering home the gut as a commensal in most all of us and in many other mammalian species, and even in non-mammalian species like birds. And so from that, I'll get started. And well, I said away from home. Well, I might as well, while you're away from my home, I just want to briefly talk about where I'm from and some of the things that happen where My institute is. So I'm in Quebec, Canada, and so Montreal is in the southern part, and Laval is just north of Montreal. And my institute is a postgraduate level institute, so we don't have any undergrads. And there are 4 centers that are divided into major themes. And you can see here that this is not English. The main language of study is French in our province. And so obviously they're international students, and it's important to have both English and French occurring at our institute. And so our institute is focused on the biosciences, so health sciences and human and animal and environmental health, infectious diseases, immunology, cancer, and epidemiology. One of our most famous labs is an anti-doping lab for athletics. So we have a group that's been involved in doing that for many years with the Olympic Committee. We have groups in toxicology, pharmacology, and obviously microbiology and biotechnology. Another interesting fact is we're also a member of the Institut Pasteur International Network, which has many centers throughout the world. From that standpoint, we are interested in global health as well. And this is an aspect that is great for international collaborations in South America, Asia, Europe, Africa. So we're very proud to be a part of that system. And it's very interesting to have collaborations throughout the world on some projects of global health interest. So importantly, I'd mentioned I'm working on extra-intestinal pathogenic E. coli, but I always never want to insult anyone in knowing full well that the pathogenic E. coli and Shigella that are associated with enteric infections like diarrheal disease across the globe are extremely important. And so it's always important to at least mention different types of diarrheal infections that can occur, and that it's something that is endemic and very problematic in Africa, India, and other parts of the world. And so although diagnostics of these diseases is not always obvious, it's still very important to realize that there are many types of pathogenic E. coli and related Shigella that are important causes of these diarrheal infections. And so that would be the intestinal E. coli. And another group of these intestinal E. coli that we know more of in the more developed countries are the enterohemorrhagic E. coli, like E. coli 0157. And so similar types of E. coli that can be associated with other infections of the gut. And so the other spectrum of infections that E. coli can cause are caused by bacteria that are essentially commensals in the gut. So that would be a normal microbiota of E. coli that can be present, that in some cases could be associated with certain kinds of chronic infection. But typically they can reside in our gut in a harmless, and in some cases maybe even a helpful fashion. However, if they are given the opportunity or somehow are able to be found in the urinary tract, in some cases, they cause neonatal meningitis, as an example. If we have surgical operations, they are often one of the main causes of septicemia as well. And it's quite different in poultry. They actually cause a respiratory tract infection. And so as well, there are clearly some extra-intestinal lesions or symptoms that can occur from some of the diarrheogenic E. coli due to their toxins. So Shiga toxins, which can cause a hemolytic uremic syndrome, and damage to the kidneys. So that just gives an overview of some of these different kinds of E. coli with the different pathotypes here having different mechanisms of adherence, different types of toxins. And so it just shows you that there's really a large spectrum of pathotypes, if you will. And so why is that? Well, here is your basic molecular genetics course when you're first learning about different aspects of bacterial genetics. And well, all of that comes into play in how E. coli has evolved over time to become virulent in different ways and in different hosts. So here you have examples of virulence plasmids, phage that can code for lysogenic phage or prophages that can code for virulence factors such as toxins, the horizontal gene transfer based on mobile genetic elements that can lead to the integration of what is known as pathogenicity islands that contain important virulence factors that can be critical for infection. So, and in some cases, there can even be loss of the genome. A change in a metabolic capacity can actually be responsible for some aspects of the pathogenesis. So this is just to globally show you some aspects of how you can have an evolution of E. coli becoming able to cause an enteric infection versus an E. coli that can cause extra-intestinal disease, such as uropathogenic E. coli. So again, the horizontal gene transfer, this is a really busy slide, but it's just to when you mention plasmids, there are clearly virulence plasmids, but it's not without saying the critical importance of the mobility and transferability of these plasmids, not only between E. coli, but between other bacterial species, and that capacity to exchange genes with these different conjugative and mobile plasmids has clearly led to a great deal of all of these red open reading frames here are resistance genes. And so this is just to give you an example of the level of resistance and the, if you will, quantum leap of becoming resistance that can happen acquiring these plasmids, which in many cases can have multiple resistance genes acquired in one genetic event. And so this has led to E. coli and other Enterobacteriaceae becoming what have been called the superbugs in the general media. And so essentially it's a major problem, and these are on the wanted list of the World Health Organization and the CDC in the States and the Canadian Health Agency as well. So I'm sure they are also problematic in the Ukraine and in Europe, and so this is of major concern. And so we have many researchers that are clearly interested in identifying new molecules or new antibiotic activities, and others that are interested in better understanding the main causes of some of these infections that we can have, and better understanding the virulence and pathogenesis of disease. And I fall more into the second category. So XPEC are bacteria that, in addition to residing in our gut, they're equipped with a number of virulence factors that can convey an ability to cause an extraintestinal infection. And so they may have certain types of colonization factors or adhesins that recognize different receptors on cells. They're adapted to overcome factors such as the iron sequestering ability of host transferrin and lactoferrin, and so they make small molecules called siderophores, which are able to chelate metals, notably iron, but in some cases they can also chelate other metals like copper. And so in addition to that ability to gain iron, which E. coli, regular K12, and commensal not equipped with such genes is unable to do. They are unable to survive in the host bloodstream for a number of reasons. There's also an ability to resist serum complement due to capsules and certain kinds of LPS or O antigens. These bacteria can often make different kinds of toxins as well. And so these toxins can allow the release of nutrients. They can also influence or affect the host immune response and make it less able to clear these infections. And so this series of factors can vary depending on the type of disease and host, but globally, many of these subcategories are conserved in E. coli, either causing infections in poultry, such as chickens, but also those causing neonatal meningitis or sepsis, and also urinary tract infections, notably in women. So there's an overlap in some of the clonal groups or the clonality of E. coli that can cause certain types of infections. And so clearly there are certain groups that are only causing urinary tract infections in women, or in some cases men as well, that are not commonly found in poultry, but there are others that overlap. And so from this standpoint, it's predicted that there may be some food associated or zoonotic, so animal sourced extra-intestinal pathogenic E. coli that can be a factor that could potentially go from the food chain into our intestines, and then eventually potentially lead to us having a carrier state that could lead to a neonatal meningitis infection or a urinary tract infection in certain people carrying these particular clonal groups of E. coli. And so, and in the avian species, well, you get some also specific pathotypes or ST sequence type groups that seem to be more specific only to the poultry, but less associated and therefore unlikely to be able to cause human infection. So there's a spectrum of broad host specificity and some that may be more specific to certain hosts. But of course, E. coli as a whole is able to cause multiple infections in different hosts. So focusing on the urinary tract infections, it's important to realize that in the case of what you would call infections in the community, so people that are generally of good health and then suddenly acquire an infection, E. coli are about 80% or so of such infections in most hospitals settings throughout the world. There are obviously other kinds of pathogens such as Klebsiella, Pseudomonas, Proteus that can cause urinary tract infections, but the large majority are caused by Escherichia coli in the community acquired disease, and in a nosocomial setting, so in hospitals, that amount is reduced considerably, but there's still in many cases 40 to 50% of UTIs can be due to pathogenic E. coli in the hospital as well. In the case of the avian pathogenic E. coli, it's important to know that these APEC are a major economic problem for the poultry industry worldwide, causing respiratory infections. When we rear the chickens, it's in a high density. So there's a likelihood, if there is a pathogenic strain that gets into the respiratory tract, it has more of a likelihood to cause disease throughout a production facility and a rearing facility. And so it can cause relatively high levels of, some cases, mortality, but morbidity and lack of an interest to eat, which eventually will also cause issues of lowered weight gain and condemnation of the meat as well due to lesions in the carcasses. So it's important for me as well, it's interesting that this is a natural host. So when you're studying pathogenesis and you are interested in a disease, in many cases, obviously for human disease, we have, cell lines and organoid-based models in some cases, but animal models are different animals. We're working on certain cases, humanized mice and things like that. Some cases, the pig may be an excellent model. In this particular case, it's the natural host for some of these strains I'm interested in. So that's a bonus for me when I'm interested in that. And so that combination of having a natural host as a scientist, and also the fact that it's a very important consumer product, and there's an interest from economically from poultry production and economically from the government as well, to make sure that we're having safe and good quality food and efficient production. This allows me an opening, obviously, to obtain research funding for these kinds of scientific endeavors. So I'd mentioned colonization as one of the first and important steps of causing an infection. And here's a slide showing you some fimbriae, which are filaments on the cells, different kinds of gene clusters that have been identified among either the enteric or extra-intestinal pathogenic like UPEC. And so you can see that there's been a tremendous amount of horizontal gene transfer in E. coli. But the first part of my talk, I'm going to focus on something that is actually present in even commensal K-12, but also in urinary tract pathogenic and also in some of the diarrheagenic E. coli, called type 1 Fimbriae. And so because it was found everywhere initially, not everyone considered it to necessarily be an important virulence factor. I'm just doing the overview of the two factors. So that's going to be something conserved in E. coli I'm going to talk about. And I'm also going to talk about a unique region in a particular strain that we identified on a virulence plasmid that has some factors that we've identified that are of interest, including a new type of fimbrial adhesin, and some toxins as well that are in this case critical for this strain's virulence. So to begin with that conserved factor, the type 1 Fimbriae, they're important for colonization in the urinary tract infection model. And so what often occurs is the bacterium can be switching from a mobile state or a planktonic state to an adhering state, which in some cases can lead to biofilm formation. And so what can occur during a urinary tract infection is you will have colonization of the bladder epithelium through the mucus. And then in the case of type 1 Fimbriae, it can also lead to internalization into these tissues. And the same type of Fimbriae has been shown to be involved in colonization of the trachea in poultry and in the air sacs as well, which is an extension of the respiratory tract of poultry. So it's been shown to be important for both colonization of poultry, but critically of interest for the urinary tract infections is this ability not only to adhere, but even to invade epithelial cells. And in some cases, it's even been reported that a strain, even after antibiotic treatment, can, when it's invaded into the epithelium, fall into a quiescent state. And then actually you can have a resurgence of an infection in certain individuals where it's the exact same strain that returns, and that may due to a change in stress or some other factors that occur, as well as the fact that that strain may be residing in the gut and then may reinfect the bladder as well. So it's interesting to note that in some cases, these recurring infections may be due to an ability of the bacteria to become latent and just situated within the bladder without causing any inflammation until there is a stress or some other change in the host that may re-trigger replication. So what are these Fimbriae? And so the Fimbriae are nanomachines, if you will. So they're a polymerized structure with a large major pilus or fimbrial rod, and they are a lectin-mediated adherence. So they have a adhesin at the tip, which generally recognizes different kinds of sugars. In the case of type 1 Fimbriae, they recognize mannoside or mannose residues on different cell types. And so there are a number of genes involved and the proteins involved. This is known as a secretion family known as the chaperone-usher family. And so the chaperones and usher are involved in the hierarchical organization of the biogenesis or assembly of the structures to make sure that the proteins are assembled in a correct order in order to generate the production of these fimbrial structures. And so the FimH adhesin tip here is recognizing mannose. And then what's interesting about Fimbriae as well, you saw in the graph that I showed that there are a number of different kinds of Fimbriae, and an individual strain can even have as many as 10 fimbrial gene clusters that can recognize different kinds of receptors on cells. And but the population is going to not be able to make all of these all at the same time. So they're regulated by phase variation. And what's interesting about type 1 Fimbriae is they have a promoter that actually changes orientation. All right, so it's an inverting switch. And there are recombinases that are important for mediating that change. So when the promoter's in the on direction, we get transcription and then production of the fimbriae. And then under certain conditions, we can have a switch to the off orientation where we'll inactivate it, and then we'll have no production of this particular type of fimbriae. So there can be crosstalk between the different fimbrial families and regulation from that standpoint. And since these fimbriae are important for the urinary tract colonization, our group was interested in trying to understand what factors can control the production of these fimbriae, and are there new regulators that haven't been identified yet that could be of interest in understanding the pathogenesis linked to type 1 fimbriae, but also the whole production and secretion of these factors may also be altered by changes in the cell physiology in general. And so if there is membrane stress or different types of stresses that are detected or changed by losing certain factors in the bacterial cell, the loss of or a decrease in the production of these type 1 fimbriae may be almost a sign of greater changes that have occurred that can be even more important than just the reduction or loss of this particular kind of fimbriae. And so the recombinases that are involved are able to mediate a switch, with one of them, FimB, being involved in both being able to switch it on and off, and then FimE more specifically being a single orientation to go from on to off in the cell. And so these recombinases interplay and compete with each other to allow a certain portion of the population of cells to produce these fimbriae. And that subgroup, when it gets into the bladder, are going to be the ones that are going to predominate to adhere to the bladder cells and invade them, the ones that are producing these type 1 fimbriae. And so this is just an example of the regulatory interplay that can occur regulating these kinds of fimbriae. So there are many different regulators that are classic, well-known regulators in E. coli that can be involved in the cascade of regulation of type 1 fimbriae. And that's just an overview, and it's a review that my group had published earlier this year. And so our hypothesis is that some factors affecting the expression of this type 1 fimbriae can contribute to the fitness of XPEC and increase its adaptability, competitiveness, and ability to colonize and cause disease. And so what we did, and so this first part here is just to talk about an approach we decided to do because of the interest of having this switch mechanism for regulation. We decided to devise a way of making a transposon bank where we could look for changes in the level of expression by putting a Lux tag in front of the switch of this fimbrial system, and then integrating it into the genome of the bacterium. And so having a marker and then having this ability to read light allowed us to, by putting it into the chromosome, have a tag, but without touching the actual functional fimbriae, which are in the chromosome. So we have both a light emission here to look for a change in the switch. And then when we do the transposon bank, if we get a decrease in light, we can take those clones and also see if we have a change in the phenotype of our actual fimbriae to confirm that it's really a downregulation or an upregulation of expression of these fimbriae. So this is just a classic transposon bank approach where we've had the fusion already into the bacterium before we do random insertion mutagenesis with here a mini TN5 system. And so from that, we could get clones where we saw, compared to the wild type strain, that would have a higher or lower level of expression of the different switch. And so as an example here, we have the wild type level with the Lux fusion, and then a mutant that we knew already was able to have a decrease. Here we're looking at agglutination of yeast. So mannose are able to, the mannose receptors allow us to use yeast instead of erythrocytes or other receptors. We can just take yeast agglutination, and the titer change is telling us that there's less of the type 1 Fimbriae produced in some of these mutants. So we just obtained ones that had lower light emission and lower yeast agglutination, and then by sequencing, we're able to identify a variety of mutants. So some examples here, and what's interesting is we had some mutants, obviously we're interested in ones that had decreased light emission and then decreased agglutination titers, which is suggesting they're going to be making less of these type 1 Fimbriae, and they could potentially be attenuated. We haven't even started to look yet in some of the groups that make more of the Fimbriae, but this is also an interesting approach because in some cases, the light emission allows a better sensitivity of testing a change in level of expression that you could not see with an agglutination approach, simply because it may get saturated at a certain level of the population being switched on and become very difficult to see a change in the level of expression just because you could not easily dilute out the agglutination titer if it was quite high. We would not be able to precisely identify the fact that something may have dropped twofold by an agglutination test easily with a high level of expression in a population compared to what we can see with the Lux fusion. So the first example here was all the genes in E. coli that have a Y are unknown function genes. And so we had some insertions in this particular system. So YQHG gene, just to give an example, as a knockout where we've knocked out the Fimbriae completely as a negative control. Here we have the gene of interest in a clean mutant. After we identified two transposon mutants, we made a clean mutant and then complemented it cleanly into the chromosome once again with an AT-TN7 system that we've developed. So we looked here in rich medium, Luria or Lennox broth, but at the same time, we tested in filtered urine, human female urine sourced, and so overnight growth. And again, there was a decrease in the level of production of type 1 Fimbriae in vitro as well. And so Western blotting as well showed us that the level of Fim here in the wild type strain, we did one with the switch locked on, which means 100% of the cells are making the Fimbriae here. And then the controls here being the delta Fim. And so the level of Fimbriae in the delta YQHG has dropped significantly to the point where it's not even detected at this level when the bacteria are cultured at an OD of 0.6, which we've done here for the Western. And so also the adherence to epithelial cell lines was decreased, as you can see here. And the expression by RT-PCR was shown here to drop about 9-fold. Again, here it's in LB we're looking at. So everything here showing a decrease in the level of expression. We wanted to see if it was linked to changes in the recombinase. And effectively, we have less of FimB being made, which, if you remember, was the recombinase allowing both directions of switching. And so it's dropped considerably. And FimE hasn't dropped as much. And FimE is the recombinase that favors the on to off. So that correlates with what we're observing, where we're having more of an off phase for these Fimbriae. So then we did an infection model using the mouse model of infection via a catheter and a urinary infection. We didn't keep a catheter in it, but it's just inserted into the bladder, the ureter, and then we would do colony counts 48 hours later. And so you can see in the bladder, the colonization here has dropped about 2 log. Complementation is not statistically different. And in the kidneys as well, there's an even more important drop in the ability to infect. So this is showing us an example here. In fact, the kidney drop here is a stronger drop than you'd see if you had a Fim-only mutant. So just the loss of type 1 Fimbriae causes something similar to this. but in some cases, the bacteria, due to the flagella, get into the kidneys. And in the wild type strains, even without FIM, some of them can continue to grow. However, here we're seeing a greater effect or a drop in capacity to colonize the kidneys, which is suggesting there are other issues in this strain. Even if type 1 Fimbriae is affected, that's very important. And so interestingly, as I'd mentioned, that switch from mobile to sessile and adhering, what we see is our mutant, that is delta YQHG. The flagellar motility or swimming motility is actually higher than the wild type strain. And so when the strain is making more flagella, it's less likely to making Fimbriae. And so this correlates with that switch between planktonic and sessile or biofilm type lifestyle. We see here as well that when it's complemented, it drops back down to levels more similar to the wild type. And here an example to show when there's lots of these type 1 Fimbriae made and the locked on, the strain is almost non-motile. And so even if it has the flagella, there's been a complete switch for becoming sessile and making a lot of these type 1 Fimbriae. So another type of change that had occurred in this YQHG mutant was sensitivity to peroxide, hydrogen peroxide. So we can see here, this is an inhibition zone of the wild type. It's about 25 millimeters. And then when we look at the YQHG, it's significantly larger, closer to 28 or 29 millimeters. And so since the zone is larger, the strain is more sensitive to hydrogen peroxide. And so that is in addition to the loss of the type 1 Fimbriae, telling us that there are other aspects of the strain that are going to be interesting to study in the future. And so what is this YQHG? That's a great question. My lab is more involved in molecular genetics, and I'm going to probably be working with some partners to do some of the biochemistry or protein aspects, if anybody's interested in that sort of thing. And so it's a 308 amino acid conserved protein that is periplasmic. It has a domain of unknown function, and there are a number of proteins that have this domain that remains unknown. The only links that we know about it is we know that this particular protein is activated by a two-component system known as BGLJ RCSB, and that this regulon is also linked to motility, biofilm formation, and various stress responses. So the results that we've had on it by itself are similar to things that occur in for instance, RCSB or BGLJ knockout, where the regulon is down, and this is one of those components that is no longer made in the regulon if that regulon is disrupted. And so that loss of YQHG is a decrease in biofilm formation and an increased motility, and that is in line with what we see from this BGL RCS regulon. And so it's going to be interesting to try to figure out what the specific functions of this protein are. And so that concludes this section here, just to talk about making an interesting library and identifying a gene of unknown function that is conserved in E. coli. So this is something that is present. And what's interesting is it is not in all enterobacteria. So it seems to be kind of E. coli specific, and it'll be in some other enterobacteria as well. But it'll be interesting to try to understand in what way this system is important for E. coli, and in our particular case here, extra-intestinal pathogenic E. coli, but it may also play some roles in some other E. coli causing other types of infection. And so we're interested in continuing to identify specifically how this protein is doing what it does. Now I'm going to switch to another mutant that we identified from this screening, which we recently studied. And it was a paper that was published last year in PLOS Pathogens. And so we identified a small non-coding RNA that affected the level of production of these type 1 Fimbriae again. At the time when we were preparing our manuscript, it was known to affect biofilm formation in E. coli K12. And one group had linked it to potential virulence traits in Shigella. And so from the standpoint of E. coli K12, it was a group that was cloning in high copy different predicted non-coding RNAs and had effects on different aspects of physiology. In our case, well, we made a knockout and wanted to see what would be happening in our urinary tract pathogenic strain. And so we were interested in looking at, since we saw a drop in the level of type 1 Fimbriae expression with this mutant, what was occurring at the level of UPEC virulence. So again, as with the YQHG mutant, we did see an increase in sensitivity to certain kinds of stresses. So here we have hydrogen peroxide. So that's more of a hydrogen peroxide stress. Plumbagen and paraquat can be more of a superoxide stress. And so again, you see the control here is a mutant that's highly sensitive to all of these different reactive oxygen species generating compounds, OxyR mutant. And we see that the RIF-A RNA mutant is not quite as sensitive as the OxyR mutant, but still is more sensitive to these different types of ROS generating compounds. Here's this is an overnight disk assay, but this is a time course with an early kill with hydrogen peroxide as well. And what you can see is the wild type. It takes quite a while to start to drop, even to drop from 100 to 60% of the cells, whereas the RIF-A mutant takes a quicker drop within less than 10 minutes. It's already down to about 50% or so. So again, it is also much more sensitive quickly to the presence of hydrogen peroxide. And so we also were looking at some other strains than the CFT 73 we use, which is an arch type strain. It's always important to make sure your phenotype is not strain specific and that it could be also occurring in other strains of your pathogenic E. coli. So this is a strain, a German strain that's well known, that's been studied for years as well. And so knocking out RIF-A in that strain as well causes an increase in sensitivity to hydrogen peroxide, paraquat, and plumbagen. In some cases also, tellurite is another product that can cause stress, and we saw some changes as well with CFT 73 and with this 536 strain, showing a greater sensitivity to stresses. Also more sensitivity to osmotic stress. So here, by growing them and then placing them on plates in the presence of 0.6 molar urea, we see a significant drop in survival in the presence of urea and also in the presence of NaCl to a certain extent. So this strain, losing RIF-A, results in both sensitivity to oxidative stress, osmotic stress, and concomitantly a drop in the level of production of these type 1 Fimbriae. So we wanted to see when the small RNA was produced in collaboration with Eric Massey's group, which is at the University of Sherbrooke, which is about 3 hours or so from my institute. And collaborating with them, we see that it is a RNA that is mostly expressed towards the late exponential to the beginning of the stationary phase. And that is so when it is being turned on, essentially starting towards 0.6 and then with a maximum level towards about an optical density of 2. And so here, this is just a northern to detect it directly from the native cell as well and compare it to a clone copy to see the size of it. And so that corresponds to what we predicted and what others have predicted as the size of this particular small RNA. So what could we do to look further into how or why it was being affected for oxidative stress, osmotic stress, and a number of different factors? We did RNA-seq analysis, and then we identified over 400, nearly 500 genes that were differentially expressed, many of them being downregulated, and many of them being linked to metabolism and stress response were highly affected. And so majorly downregulated genes here. Everything in red that you see on the far left are associated with the general stress response. So these can be phase shock genes, cold shock genes, heat shock genes. A lot of those were showing a considerable downregulation. An upregulation, there were only a few factors that were clearly identified, and some of them were more linked to metabolism of sugar, for instance. And so we still have quite a bit of work to figure out exactly how or why losing this small RNA has this kind of effect on the general or universal stress response, and also in some cases affects certain kinds of metabolic pathways as well. Something that was also interesting is we noted some other fimbriae had been affected. The P fimbriae or PAP fimbriae are important for urinary tract infection, particularly in the kidneys. And we did detect a lower expression of genes associated with that other fimbriae. Both, there's 2 copies of that fimbriae in CFT73, and both of them had dropped significantly. And so this is just some RT-PCR to confirm some of the genes that we had seen by the RNA-seq that were either downregulated or upregulated, and that correlated with our results, which was very nice. And then we also wanted to look in vivo. And so when we looked in the bladders at 48 hours in the infection, we could identify that the wild type strain compared to in vitro medium, we had about 5-fold increase in the level of the copies of RIF-A being produced. So suggesting that it is induced and is a player in regulation in vivo. So we did a knockout, obviously, in vivo. We were looking at importance for it for type 1 fimbriae initially, and we showed there's lots of effects on stress. And here as well, we can see there's a drop in the agglutination titer, whether it's in medium or urine overnight, an important drop in also static medium, which favors type 1 fimbrial production, and also in urine, again, a very strong and important drop. Western as well, showing us a significant and important drop in the level of production of this fimbriae. And so the Fim expression was also decreased by looking for Fim-A under different growth conditions, such as LB, static urine, and in the bladder as well, as you can see, up to a 6- to 7-fold change here for detection of the major fimbrial subunit in vivo. So loss of RIF-A resulted in a significant drop as well there. There was also a higher level of motility, and importantly, a high level in the RIF-A mutant. So despite having some issues with increased sensitivity to oxidative and osmotic stress, clearly the flagellar system is working very well, and this bacteria is highly motile when it's lost its RIF-A small RNA. And so when it's complemented, it returns. And as a control, as I'd mentioned before, when you lock on type 1 fimbriae, you pretty much abrogate motility. So the RIF-A mutant, it's very motile. We saw type 1 fimbriae were decreased, but on electron microscopy, we still see there are still fimbriae that are made by this RIF-A mutant. And so that interested us, and it actually still adheres to human bladder cells. Even if we had a drop in type 1 fimbriae, that doesn't mean that there aren't other mechanisms of adherence that can be occurring. And so we can see that here, that this strain actually adheres just as well as the wild type. When we add manos here, this means that type 1 fimbriae are inhibited. So this wild type strain, much of its adherence is dependent on type 1 fimbriae, but the RIF mutant is likely to be using obviously something else because it's a mannose resistant adherence. When we complement, again, it falls into the profile of becoming type 1 dependent adherence. And so you can also delete completely the Fim operon from the same strain, and it still adheres very well to cells. because it has backup mechanisms, it has other adhesins. So it's not just adhering in vitro that's going to give you an answer about the importance of this small RNA. You need to do an infection in an animal to see a difference and an importance, and also know that there are changes in stress resistance that have occurred. And so if you take a human erythrocyte agglutination, however, there we've got a significant drop. So the adherence that we saw just to the epithelial cells was, in some cases, not adhesins that are likely to be hemagglutinins. Essentially, that's what we're seeing here. So in knowing the strain we were working with, we had some ideas of what Fimbriae could be present and what Fimbriae that are maybe present, but also maybe aren't known to be able to hemagglutinate human erythrocytes. And so we looked at these different fimbrial systems, and as we predicted, we saw in our results for the arrays a drop in PAP-A and the 2 copies of the PAP Fimbriae, a drop in FIM, and there's another Fimbriae called F1C that can mediate adherence to bladder, epithelial, and kidney cells. And so we saw that that was upregulated by RT-PCR. And I had a collaborator in Montreal at the vet faculty that actually worked on this kind of Fimbriae. And so she gave us some antibodies against it. And sure enough, we see it's actually increased in expression, this F1C Fimbriae. So even if type 1 is down and P are down, the bacterium is altered in its state and more stressed to certain conditions. It's still making another kind of Fimbriae and actually making more of it than it would in the wild type. So again, the virulence is the last type of experiment we want to look at in the animal model. So we see a strong drop in the bladder of more than 2 log, and an even more marked change and importance in the kidneys. Again, this is not surprising if the bacterium is also more sensitive to stress. And again, the complementation, which was done by integration single copy into the chromosome, results in a nice clean regain. And so looking more at the host pathogen interactions, we looked at also the ability for the bacterium with loss of RIF-A to either be internalized or survive in primary monocytes, human monocytes, as well as the THP1 macrophage cell line. And so what we can see is we see that there is a drop in the ability of it to be internalized by the primary monocyte cells, and also survival that remains low over time when we do the incubation of these cells. The complementation results in a regain of the ability of it to be internalized and also to survive in the primary monocytes. The results are slightly different for the THP1 cell line, but what we still see is that although it seems to enter just as well, the RIF-A mutant, it is less able to survive over time. 2 hours later and 24 hours later, there's a significant drop in its ability to survive in these cells. Again, the control that we used here is the highly sensitive OxyR mutant, which is very sensitive to oxidative stress, and which explains why it's not having a very good time trying to survive within a monocytes and a macrophage cell line. So the conclusions on the RIF-A are that the deletion of this small RNA had important effects on E. coli stress resistance and virulence traits, and also expression of different kinds of Fimbriae. And it's going to be interesting to identify what are the specific roles of RIF-A to either directly or indirectly control these close to 500 different genes that we identified in our initial study. So the mechanisms of it. So a quick last little section to go into the genes that are more pathospecific and the avian pathogenic miles. So some of the people that are more interested in veterinary aspects will maybe appreciate this a bit more, but it's also interesting to understand that there are virulence factors that are pathospecific. I talked about type 1 Fimbriae, which are in most E. coli, And the two regulators we identified were conserved genes that are also in E. coli K12. Here we're going to talk about genes that are very distinct and pathospecific and in only certain types of pathogenic E. coli strains. And so E. coli infects the air sacs, which are important for thermal regulation of poultry and which lead into respiratory or aerosol infection of the lungs that can after that cause systemic infection. And so you can get diseases like pericarditis and perihepatitis in these animals, which leads to great loss of production and loss of meat in the poultry industry worldwide. Now, one of the factors that is really important for survival, as I mentioned, are these iron chelators. And so I just wanted to point out there are 4 known E. coli and Salmonella siderophores. And it turns out that enterobactin here is conserved by E. coli K12. What you can see is salmonellans are nearly identical. However, they're glycosylated. So Salmonella and some E. coli that have these salmonellan genes have trafficked or altered the the base enterobactin synthesis pathway to modify this molecule and make it glycosylated. Aerobactin is a third factor, which is a hydroxamate siderophore, which is very important in many cases for survival in the blood and different types of extra-intestinal infections. And Yersinia-bactin is the 4th member that can be present in some strains and can be important in iron sequestering, but has of recent years been also shown to be able to sequester copper as a defense for XPEC as well. And so most enterobacteria have enterobactin, as I mentioned, and then this is a breakdown of what we see with the presence in so some APEC or XPEC, it's on virulence plasmids in many cases, the colicin V type virulence plasmids, which are present in nearly all APEC. Just to give you that overview. The reason why I'm bringing this up is because I actually, when we were working on these metal transporters, we were even using mass spec to try to detect these different molecules. And we found some strains that didn't make aerobactin, but they had the gene cluster. And so we wound up sequencing one of those strains, and that led us to some interesting findings. And so just to go and explain to you briefly, why would you add glucose to enterobactin? And it turns out that, interestingly, people working on a host defense response will be aware of a protein in inflammation called lipocalin, which is also known as siderocalin. And when we were doing research in the host defense aspect of proteins, it was actually identified when somebody did the crystallography of lipocalin-2, or also known as NGAL, that they cloned it in E. coli. And when they did the structure, they wound up finding that there was actually a molecule that was present within the structure, and it turned out to be enterobactin. And so it turns out that this host defense protein can sequester the iron sequesterer of commensal E. coli to neutralize it. And so to circumvent that, and that's actually detected as a pattern recognition sequence by some bacteria and can trigger IL-8 production. And so it turns out that when you glycosylate this molecule, it's a means that's allowed Salmonella and some pathogenic E. coli to circumvent the host defense response. And so make their iron sequestering molecule impervious to the capture that siderocalin or lipocalin-2 is capable of doing. And so this makes salmonellans important. And in my own lab, we actually cloned and found the copy of the equivalent NGAL that's present in poultry. So the same type of system can exist in poultry as well, where there is a siderocalin-like molecule. It's quite different, but still it's able to, and I don't have time to go through it to show you the biological results showing that it can sequester iron from E. coli K12, but we published that a few years back. And so we found this strain that had something wrong with aerobactin, which has typically been shown to be an important siderophore for virulence. But this strain also had a unique region that was missing. This is the salmonella system. The salmonella system is in both of them. And then certain genes are shared, but this region here was different. And it turned out it had a P. fimbriae-like operon in it. And fortunately, we just got the paper out in Applied Environmental Micro. It was actually preprint published yesterday on this fimbrial family. There's an autotransporter and a toxin, which is an RTX-like toxin, right in this region, all three in one shot. And so are they functional, and can they contribute to virulence, and are they important for systemic diseases in poultry? So just a quick overview to wrap up. The autotransporters are really neat molecules. They are serine protease autotransporters. They can mediate adherence. They can also have enzymatic activity such as mucinase and hemagglutinates. And so they have multifunctions. They can be present on the surface, as you can see here from this electron microscopy immunogold image. And so they're called autotransporters because they make a pore to self-express themselves on the surface. Since then, it's been found, obviously, they have partner proteins such as the BAM complex that are necessary for this presentation. But this particular new member, our strain that we were interested in, actually was found to have 5 different autotransporters like this. And so it does function as a hemagglutinin. And so these are 3 of those different autotransporter proteins, 2 of which are on a virulence plasmid, and the other one is in the chromosome. They are very large proteins, and they have multiple activities that can include adherence to erythrocytes and potentially mediation of adherence to host cells, and even a function potentially as hemoglobinase in some cases. The fimbriae that we saw in that operon, are they functional? We did see that they can be expressed when we clone them into an E. coli strain that had no fimbriae. And we also saw that their new hemagglutination activity, unlike true P. fimbriae, they can also agglutinate not just human erythrocytes, but actually turkey erythrocytes. And the strain was originally from a turkey. So we found that this is a new kind of a P-like fimbriae in our strain, and that it's present on a virulence plasmid that's also mobilizable and so conjugative. And so that's quite interesting. There you can see them here at higher resolution. Finally, there's this RTX toxin as the third region. And so this is just when we clone it, we can see it's able to lyse erythrocytes. In the wild type strain, though, you can't detect that. There's no hemolytic activity. It's silenced in vitro. And so when you clone it in a higher copy, you can detect this functionality. And so just to see, you can see clearly that when you have this toxin produced, it's very lytic to cells, and whether it be avian cells or human cells, we saw that it was able to form pores and kill cells. So we did an infection in the turkey model, 3-week-old model, and essentially we're looking at, from air sac infection, whether there's colonization in the extra-intestinal tissues. And so we tested a number of strains, and what we wanted to do here was we had the strain that had the original col-V plasmid with aerobactin, and which does not have this unique region. And then we had a plasmid that we could use that had lost the genes from this unique region, so what we call the enhanced virulent island, and then a plasmid that completely was cured of the virulence plasmid. And so in order to let you look at the difference of the importance of one plasmid versus another. So the wild type strain has the traditional col-V plasmid. When it loses this plasmid, it's highly attenuated and unable to grow in the blood. Here we've switched the plasmid to the one that has the unique virulence island, and you can see it's more virulent, it's getting higher numbers in the blood, and then here we actually had mortality where we had, with this new plasmid with the unique virulence region, we had 6 that had died by 48 hours. So it's actually killing them compared to the wild type strain with the conventional plasmid, where we had 1 death and just 2 by 48 hours. Here we had up to 6. And so in the tissues, again, we saw with the virulence plasmid that has this enhanced region, and then when we dilute or delete that unique region itself, we wind up having a drop, a major drop. So this is showing us that on this virulence plasmid with the unique region, those 3 systems that are present are extremely important for this capacity to colonize the lungs, the liver, and the spleen. And so this tells us that, you know, traditionally the APEC strains that have been studied, these 3 regions have not really been discovered before. And so it was quite exciting for us to study that change. And so these are emerging new pathospecific virulence factors. These are just lesion scores in the air sacs to show again that there's a critical importance for this unique region in causing disease. And so to conclude, for that, this virulence plasmid that lacks aerobactin, which is known to be very important for many kinds of extra-intestinal infection, can contribute to virulence and systemic infection, and it can actually even be increased, and that this enhanced virulence island locus with these 3 different virulence factors confers an increased virulence and disease, and that this strain having this particular plasmid does not require the aerobactin siderophore for virulence. And so what's interesting as well is, since this plasmid is conjugative, it's possible that over time there may be, the same way we see transfer of antibiotic resistance, there can be emergence of new pathotypes and changes in virulence that may occur if the genes from this plasmid get transferred into different backgrounds. And if you do a genomic search on them, which we did in the paper that we just published for the P. fimbriae, there actually are a considerable number of human infections with strains that are carrying this Colby-like plasmid with this unique region, or this enhanced virulence island region in them, associated with human sepsis and urinary tract infections, as well as the presence in for instance, dog urinary tract infections, and obviously a presence in some poultry samples. And so to conclude completely, pathogenic E. coli have inherited and exploit a variety of virulence factors to cause different diseases. They likely continue to add new virulence and antibiotic resistance mechanisms to their survival playbook, and they continue to be a challenging pathogen for humans and other animals. And so as part of that playbook, I take-home message, I presented the importance of conserved genes as an example with regulators of type 1 fimbriae and urinary tract pathogenic E. coli, and also the importance of distinct pathospecific virulence genes on a transmissible plasmid that can increase virulence in poultry as well. I'd like to thank members of my lab. In particular, the main reason I'm here is actually my contact through Katerina Krylova, who was an excellent postdoc working on a project with bacteriophage, where we did some good advances, and we still have a manuscript that should be coming out pretty soon with Katerina as a co-author. And with that, I thank the funding agencies I have, and I also thank you. So it's Dia Kuyu Toby, right?
Yes,
thank you.
Thank you very much for the very informative lecture. And we have some questions in chat.
Great. I'm very happy to answer questions. I'm going to stop share, maybe it'll be easier to see. Whoop. Okay. And in chat, I have, that's a great question. I think it's interesting to think about that too. And Nick has a question. So how long ago did salmonella evolve? And why do not all pathogenic E. coli use this trick? It's in all Salmonella. So, and that's why it's called salmonellans. And so in Salmonella, it is a pathogenicity island that is present in the chromosome. And in E. coli, in some cases, it's on the chromosome. And in some cases, it's on these virulence plasmids that are these col-V-like plasmids. And that's also been inherited in some cases by some Klebsiella strains. So some enterobacteria, I guess, that have acquired it, have become able to cause extra-intestinal infections. And from that standpoint, I'd say it's probably, it's got to be relatively old in Salmonella, as old as Salmonella probably, because it's in all Salmonella branches. But yeah, and I think that in other cases, it may be because some of the other E. coli have the other backup siderophores. So they can have aerobactin or Yersinia bactin that can compensate and allow them to get iron. And then finally, there's some E. coli as well that can use other sources of iron because they have maybe a hemolysin and then heme transporters, for instance. So there are some that won't need a siderophore, and they'll be able to sequester iron through heme or hemoglobin.
Thank you.
You're welcome. I've got Konstantin has got his hand up. And there may be some other chat questions, but I'll see.
Yeah, I have a question. Thank you very much, Charles, for the great presentation. I have a question about the simple question, but very important.
Sure.
From about the domestic.
Your audio cut out on me.
Sorry.
What is the first step you should do when you cook eggs, chicken eggs? You should firstly wash it, or you should firstly boil it to prevent salmonellosis or shigellosis.
Well, that's a good. I actually have 3 chickens at home in my backyard, so I don't just do that at a professional level. It's never a good idea to wash an egg because you're actually going to, the pores in the egg can actually sequester anything on the surface when it gets wet. And obviously when you boil it, you're fine. But if you were to wash it and then, use a raw egg in some kind of, you should never do that. There's always a risk, but boiling it is important. But if you wet it, you're with water and it's not boiling, you can actually have that temperature change and have the same thing happens with E. coli in like lettuce, like you can wet it. And then in some cases, if it's not washed properly, you can actually get O157 inside the pores of the plant. And so that's an important question. I think if in certain parts of Europe, eggs are clearly not refrigerated, and that doesn't mean they're not, they're a risk when they're refrigerated as well, in any case. But it's a good question.
You give advice to firstly boil eggs without any washing.
Yes, or I mean, if you're going to fry them or something, obviously you're not going to need to boil them. You can just crack them open, and as long as they get cooked well enough, you're going to remove your risk of infection and contamination. But your point, I think, is interesting as well, because some of the E. coli I talked about here today, I didn't talk that much about Salmonella, but there's probably more APEC-like or pathogenic E. coli in some cases on our eggs than Salmonella. And so we may be, in some cases, when we're eating poultry-derived products or eggs, increasing our likelihood of contaminating our gut with potential pathogenic E. coli. But if they're only causing extra-intestinal disease, we're not going to realize it easily. You know, so somebody can get an E. coli that's going to deplace other E. coli in your gut, and then maybe 3 weeks later, a woman can get a urinary tract infection, and the source of it could have been a contaminated egg. But it's not easy to do the epidemiology to that. It's much easier to identify, oh, I got Campylobacter, or I got Salmonella because I had uncooked poultry. You know, within a few days after eating it, you're going to be ill, and you may put that together. But the idea of having maybe some pathogenic E. coli, which is often present in higher numbers on these samples, is less obvious, because in the gut itself, these bacteria are not causing disease in general. I mean, if you read in detail about APEC, you're going to find some information about Crohn's disease, and even some papers on things like there's a colibactin that's made by some of these bacteria that can be associated with cancer in the colon. So there are maybe some E. coli that are, you know, APEC-like, that are in the gut that can be causing chronic problems. I don't want to not mention that. I just want to make sure you know that not all of these APEC in the gut may be completely friendly, depending on the human host, and if they have other factors, you know, the Crohn's type and IBD and things like that. Some of these types of E. coli can be much higher presentation of them. If you have some talks on microbiomes and things like that, you'll be hearing people that may mention some types or pathotypes of E. coli that are associated with gut infections, and a lot of them are APEC-like.
Okay, thank you very much. So I will
Thank
boil eggs before washing and after wash.
I recommend it. I mean, sometimes if you have recipes where you have a raw egg, if you know the source of your eggs, and if you've had little issues, but I think it's mostly very important, particularly if you have young children or elderly, to make sure that your eggs are well cooked to reduce any risk of having problems with people that may be more susceptible in particular.
Thank you very much.
You're very welcome.
Please ask questions.
Okay, I'm going to see in the chat.
Yes,
Okay,
I have some
I've got a lot of
questions.
thank yous. I'm going to maybe backtrack a bit, but I've got, let's see, good. Does FIM have a role to play in eliciting hemolytic uremic syndrome disorder? Not that we know of. It's basically the Shiga toxins that are produced by the E. coli that are either they're called virotoxigenic or Shiga toxigenic E. coli. And so these are encoded by prophage that are integrated into the genome. And this point is very important because initially, before people understood the mechanism of disease of EHEC, the use of certain types of antibiotics could actually be very detrimental to the patient. So as an example of that, if you take a fluoroquinolone type antibiotic, which is DNA damaging, everyone in your institute and quite a few people that are experts with phage will know that when you have DNA damage, it's like the SOS response, and you're going to get prophage activation. And so when that happens, you get extra production of the toxin. And so in some cases, initially, before they knew the mechanism of the disease, if they gave an antibiotic that was a DNA like ciprofloxacin or something like that, you could kill all the bacteria, but you could kill the patient, because there would be a huge amount of toxin made as the phage were replicating before the bacteria lysed. So it's not type 1 Fimbriae, it's Shiga toxins. Was perihepatitis made in chicken with injections or E. coli migrated to the liver? In the case of our infections, we use an air sac inoculation route. So the bacterium have to go from the respiratory tract. So I do a direct injection into the air sac for uniformity. The natural route obviously would be an aerosol infection. But what would happen if we did an aerosol infection is you might get, one bird in 3 that might get enough of a bacterial infection to develop. And so in order to have a more uniform infection, particularly in the bloodstream, we do an air sac inoculation, but the bacteria have to be able to disseminate and survive and resist complement, for instance, to get into the blood, and then to be found in either the liver and also the spleen. Okay. I think that looks like 2 new messages. Okay. What was the matter to induce UPEC in mice for 48, but not for 24 hours as a chronic disease? Okay, that's a great question. Different groups have done different models. A lot of people consider 48 hours to be still kind of acute as an infection. In some papers I've written, and a question came up as well in looking at the late induction of RIF-A, for instance. I had some reviewers ask, why didn't we go to 5 or 7 days even in some cases? And it can depend on the mouse model. Some are more susceptible than others, and some may clear it by a later time point. 24 hours I've done a few times, mostly when I was looking at taking a subgroup of infections and looking at the kinetics of expression of a particular factor. But I've never really compared 24 versus 48 hours. But I've had good counts by doing a 48-hour infection. The animals themselves don't appear to have any major stress or issues by 48 hours. So, and we still have a pretty good amount of counts in the wild type, which allows us a good comparison for any mutants that seem to have been attenuated or have dropped an ability to colonize. But no, it's really, I think it depends on the strain you use as well. People may get different results with different strains, and also the line of mice. I didn't mention that, but in our case, we're using CBAJ mice, which are pretty much a general line that people can use. There's no issue with NRAMP or anything like that in the case of these UPEC infections, unlike Salmonella or Mycobacterium, where you need to use a BALB-C sensitive line versus other lines. That's just my feeling on that. Yes, of course, I'm planning to continue to do research on E. coli. I'm a young 53 years old, so I'm still happy to be doing research and continue to work on different aspects of pathogenic E. coli. And some of the things I talked about today are still in my cards for future projects. I'm also interested in maybe trying to do a bit more work with different animal models that we found that could be of interest to do studies on pathogenic E. coli, which can be as varied as the zebrafish model, and also looking at turkeys, because as you saw, some of our adhesins seem to recognize turkey erythrocytes, but not chickens. So, but still cause potentially infections in humans. So yes, my answer is yes, I'm continuing and interested in that. What is my opinion about the therapy with enteropathogenic bacteriophages? I think that's a great question. I don't know if you had Rob Levine talk yet. He's later in your sessions, he'll be talking. He's actually at this meeting with me here, and he gave a great keynote the other night. I think for enteropathogenic infections, if you're able to have a cocktail, and you're able to have a way of ensuring that your phage are well protected from the acidity of the gut. Obviously, you can use some buffers and certain things. Some types of enteropathogenic infections can be quite well treated. I know with Shigella, there are many cases where it's been shown that it can work quite well. I think that a lot of the interesting aspects that have been shown so far are problems where they're chronic infections and people that have things like staph infections and medical devices or hip replacement and things like that seem to be an area where bacteriophage are really the only thing that can work, because antibiotics, they just can't get into the pharmaco distribution of the drugs don't seem to get in as well as in some cases the bacteriophage can suddenly infect some cultures. So I think phage are very interesting, and I think there's lots of potential. But at the same time, there's lots of questions, and people are talking about bioengineering, and you can do synthetic biology, and there's so much potential with probably phage proteins as well, things like endolysins and alternative to antibiotics like that, where we could have tailor treatment if we have the right diagnostics for certain kinds of infections. I think that would allow us to be much more judicious in our use of treatments, and that could in itself lower antibiotic resistance across the globe. So there are things I think we can learn from phage as well, not just by having them as full viral bacterial killers, but also by the molecular biology of some of the proteins they make, and that can have super interesting capacity as well, maybe as future ways of alternatives to antibiotics as well. Yeah. Okay. So yeah, your answer, Yulia. I think if your model works at 24 hours, great. I mean, it's the whole point is if you're trying to do comparisons, that's what counts. I went to Jim Johnson's lab, who worked on UTIs for years, and as a postdoc coming out, starting my lab, he had a 48-hour time point, and I stuck to it. And I mean, if you do another model and it works, then that's great. As long as you have good controls and good statistics, more power to you. I think it's great. And depending on what you're looking at for, if you're looking at the host immune response too, it may be more interesting for you to have earlier time points, or even to take, time points in between your final time to kind of look at, the development of an immune response and things along those lines. Yeah, people who have urinary tract infections, there are some people that unfortunately they can have recurring infections, which leads to evidently multiple usage of antibiotics, which can unfortunately cause issues such as perturbation of the microbiome, and then cause problems like inflammatory bowel disease and Crohn's, and they can also develop things like yeast infections. So this is a terrible thing for people that have, in some cases, a reflux issue or are more prone to be infected. And so I think that it's very important to try to be as careful as possible to avoid a cross-contamination, fecal to urovaginal cross-contamination, and to make sure that you drink lots of fluids. And that way, hopefully, you're going to be able to be urinating regularly, and that flushing and refluxing is one of the main ways that every and any one of us can lower the risk of infection. And so, in some cases, it can be people that are less if you're urinating less often, you're going to increase your likelihood of potentially retaining an infection. and then obviously, you know, there are products that are projects underway for different groups to, you know, potentially develop vaccines and things along those lines. We'll wait and see, but it's not clear at this point that something like that can occur. And admittedly, someone that also has an anatomical propensity to get an E. coli infection can also get other kinds of urinary tract infections, most likely. So even if we did have an E. coli vaccine, some of those people, I think, unfortunately, would suddenly maybe get a Proteus infection or you know, another type of bacterial infection potentially. So I think it's a tough area, but you know, there are things, at least drinking lots of fluids, and you know, there are theories that potentially drinking things like cranberry juice or products with lots of mannosides may somehow lower adherence. I don't know to what extent, though, how much mannoside or mannose residues actually get into our urinary tract. It's quite doubtful, because these are good sugars we like to use as a metabolic source, so it's not a waste product. So it's unlikely that we're getting a lot of sugar unless we have issues with diabetes or things like that, which are also a group of people that are more susceptible to UTIs in many cases. Can I do some work with phages? I have to see if my phages are still functional. I can tell you too that as a sideline project, I probably can't necessarily share them with you, but we did. I do have some from my own stuff I did with my group that were looked at more from a poultry project, but we did have a side project that I'm still maybe trying to wrap up, where we did have some phage that came actually from Cote d'Ivoire, with a collaboration with someone from Africa. And I think it's interesting, and I think that it's important to collaborate, and if groups and people, for yourself in Kenya, if you do actually isolate some plaques or get some clean phage, I think it's important to collaborate if need be. There are lots of labs doing sequencing, and together, I think it could allow you to properly analyze the phage and make sure that they have no genes that can lead to temperate integration, and no virulence factors and no antibiotic resistance factors, and you'll see how important that is when you're able to maybe select potential phage that could be useful, possibly for phage therapy in some cases, which may be of interest in certain parts of Africa and other parts of the world where there may be an increased risk of enteric infections due to water quality or problems accessing clean water, particularly depending on the time of year where there may be issues with overflow and flooding and contamination of river sources. I'll take your email down, and anyone else, obviously, if you have other questions and you want to contact me, please feel free. I'm pretty sure my email is on the beginning of the recording when we had the session recorded, and if not, well, I'm on the Facebook page for the summer school, and feel free to send me a messenger thing from Facebook, and from there we can talk about different aspects of things that might interest you.
Thank you very much, Professor. It was very interesting lecture, a lot of questions, and it was very nice to hear a lot of material.
Yeah, and I mean, to close, I have no option but to also be political and say I'm very angry about what's going on to you people.
Yes, unfortunately, yes. Today we had several dangers from air signals, but we continued our school because sometimes missiles are flying above our city and other cities in Ukraine, but we try to be brave.
Yes, and you know, science is international, and everybody has to work together. And so I don't understand when countries do things like this. You know, Canada's a country that sort of just came to be because, you know, the Brits got bored of us almost. We didn't have a revolution. We had wars within our country, but you know, and we fought for wars. Obviously, you know, in World War II, our nation became a nation essentially by going to Europe and helping in World War I as well. And we understand that these things can happen and have happened in the past, but I personally don't understand how they can happen in the 21st century right now. I want you all to know that I'm with you in spirit, and I've sent some donations to certain causes. I know we need to do more though, and it's, you know, as far as I'm concerned as a scientist, if there's any way, if there are some of you that are interested in the type of research I'm doing, and if there's a way of having funding that can be you know, somehow also favored for people coming from the Ukraine and that are interested in continuing their research. You know, I'm certainly very proactive in trying to have people come to my lab if they're interested. And I just want you all to know I'm glad you're having this format and you're getting a chance to speak to many people across the world. If the COVID situation has allowed us anything, it's this ability to actually, you know, decide to have speakers from all over the world, and us realizing that we can communicate through Zoom and not have to get on a plane. And it would be nice to see people in person and go out to dinner and talk, but at least we can do this. And I think for your situation right now, it's even more powerful for you to be able to have a chance to do this. And keep up your good spirits as much as possible. And I'm looking forward to a resolution to what's happening over where you are, and hopefully continuing economic sanctions and other pressure. I'm hoping that you're going to be able to be incorporated as a member of the EU eventually, and that things along those lines are going to strengthen your likelihood to continue to have your solidarity and your independence as a proud nation among us. And with that, I hope you enjoyed the science. And I wish you all an excellent continuation for your summer school.
Thank you very much for your kind support and for your kind words and support and help. It means a lot of us. Thank you very much.
Great. Take care, everyone, and all the best in all your studies and projects.
Thank you. And maybe some participants will communicate with you after, because maybe a lot of people are interested in your research.
Great. It was a pleasure speaking with you. It would have been nice to see more than postage stamp size faces or black screens, but I hope you at least appreciated what I had to talk about, even if we weren't face to face.
Yes, sometimes we have no very good connection. So if internet is not very high speed, so we should.
But in my case, everything was pretty good. It didn't cut out, or?
Very good. It was very good.
Great. I'm very happy. I was a bit worried being at a hotel that I might have some issues, but great. Okay, take care. It's all ready for you guys. It's almost, I guess, you're looking at what, 4:30, something like that. You're wrapping things up pretty soon, I guess.
We hope for the victory, for better times. Yes.
Yeah. Okay. Take care, everyone.
Thank you so much.
My pleasure.
Thank you so much.
Bye-bye.
Bye. Dear participants, thank you very much also for your kind questions and for your support for Ukraine, also for people from Kenya and other countries. Thank you very much for your kind support for Ukraine. And I was very glad to communicate with you today on summer school, and we will meet each other tomorrow. Thank you very much.
Thank you. Goodbye.
Thank you very much. Have a nice day. Goodbye.
Have a nice day.
Thank you. Goodbye.
Bye.
Thank you. Have a good day.
Thank you. The same for you. Thank you. Thank you. The same for you.